Hello, I'm Dawson, a postdoc in the breast cancer lab of Jane Visvader and Geoff Lindeman at the Walter and Eliza Hall Institute of Medical Research in Melbourne, Australia. I just finished my PhD where I was working on breast stem cells and immune cells using 3D confocal imaging and intravital imaging. So today I'll talk to you about how these led to new insights into resident macrophages of the mammary gland. Please get in touch by Instagram or Twitter so you can ask questions and we can discuss this. The mammary gland is a really dynamic organ that remodels a lot over the life of a female mammal. So I'll introduce you to this, and then I'll introduce macrophages as well. These are really diverse immune cells that have very specific functions in every organ. Then I'll show you how we made a really interesting observation using 3D confocal imaging of mammary ducts spying these new immune cells, and then go through how we characterized these and found that they were a macrophage population. And then I'll go into some studies in breast remodeling and cancer to see what these cells do. So the mammary gland is a really fascinating organ because it primarily develops after birth. This diagram shows the development of the mouse mammary gland. At birth, there is just a small ductal tree, and this doesn't grow a lot until the beginning of puberty at 3 weeks, when terminal end buds form at the ends of the ducts, and these house proliferative stem cells which drive duct growth and branching throughout a fat pad. And this generates an elaborate ductal tree. In pregnancy, this tree blooms to generate spherical alveoli, which produce milk. And after lactation, in a process called involution, these milk-producing cells die and are shed, and this returns the gland back to its resting ductal state. So our lab, we use 3D imaging to image the different stages of mammary gland development, and we use whole mount imaging of cleared tissue. And this shows us the shape and the architecture of the tissue, as well as individual cells, where they are and how they interact. The mammary ducts are made up of an epithelial bilayer with outer layer of myoepithelial cells that are contractile and elongated, and an inner layer of luminal cells which mature in lactation to produce milk. And with the 3D imaging, we can get a big picture of the stroma that surrounds the duct, as well as the epithelial cells and any cells that we interact with. We use a clearing agent called fungi that was developed by a previous postdoc in the lab named Rios, and this was published last year. Tissue-resident macrophages are a really interesting immune population. They're really abundant in all tissues, and they develop from monocytes under the action of the CSF1 growth factor. Macrophages typically express F480, CD64, and MERTK, and we use those markers to isolate them. Traditionally, macrophages are split into M1 or pro-inflammatory macrophages and M2 or anti-inflammatory macrophages from fairly typical inflammatory or anti-inflammatory signals. But this is a little bit simplistic and applies mostly to in vitro pathogenic situations. Whereas under homeostatic situations, a vast variety of factors programs resident macrophages into a really diverse range of phenotypes and functions depending on the tissue and the tissue niche. But some universal functions of macrophages are in tissue maintenance and repair, phagocytosis of pathogens and apoptotic cells, and regulation of immune responses. And here are just some examples in different organs. We have individual or even multiple macrophage populations, each with very specific functions for that organ. Some of the macrophages have a lifespan of the life of an animal, whereas others survive only for days, whereas others for months. different populations of macrophages have different origins. They're first generated in the embryo from the yolk sac and the fetal liver, which seed all organs in these first waves. And in organs like the brain, which develops the blood-brain barrier, after these embryonic macrophages seed the brain, they are not replaced and they survive throughout the life of the animal. Other populations, however, lose their embryonic macrophages over time, and they are replaced by bone marrow-derived macrophages in the adult, such as in the intestine. It's not really clear yet what the differences are functionally between adult and embryonic-derived macrophages. So what do macrophages do in the mammary gland? Well, in puberty, as these ducts grow and branch throughout the mammary fat pad, the macrophages are required for this growth and branching. And they sit within the terminal end buds where they clear away the apoptotic cells. We know that antigen presentation negatively regulates this growth, but it's not clear whether this is macrophages. In the adult, macrophages promote mammary stem cell activity, and they closely interact with the epithelium. And there are some persistent embryonic macrophages in the gland. In reproduction, they regulate alveogenesis during pregnancy and are really important for the remodeling that happens in involution. But we really don't know much about the heterogeneity of macrophages in the breast tissue yet, and we don't know by which mechanisms they carry out these functions. So the reason that I'm telling you this is that early in my PhD, I was looking for different types of cells that interact with the mammary ducts. And one of these was immune cells. So this is a 3D confocal image with the mammary duct here and immune cells in yellow. And looking at an optical section, you can see this immune cell stain within the duct. And looking at a thicker section, these start to take form as these dendritic shaped cells that are within the epithelium. Looking closer with more specific stains for each of the epithelial layers shows that these immune cells are sandwiched between the two layers of the epithelial bilayer. So here's the myoepithelium, and here's the luminal layer with the lumen of the duct here and the breast stroma up here. And we were able to look at different markers for these cells because they have such a specific localization. And so here is the CSF1R, very typically expressed by macrophages, which is positive in these cells, and positive for CD11C as well, and negative for CD11B. And so this lets us hone down on these cells and try to figure out how we might be able to isolate them. And these markers are actually quite unusual for a macrophage population, because most macrophages are CD11B positive and CD11C negative. Doing some rendering in Imaris using the opaque signal and cutting off digitally the front half of the duct, we can see these cells create a network underneath the outer layer of the epithelium. And when we image in lactation, we can see these cells still, and they interact really closely with the myoepithelial cells, which are stained with a keratin 5 or keratin 14. And these myoepithelial cells wrap around the alveoli and contract to squeeze out the milk. They also seem to be present in human breast tissue. We have these cells just underneath the outer layer of the duct. Other people have spotted intraepithelial immune cells in the breast before. Here are some, and some with CD11C, and then others more recently associated with the myoepithelial cells and the epithelium. And then here, macrophages underneath the outer layer. But it hasn't been shown whether these cells are a distinct population and whether they might have important functions. And so using the information that we got by imaging, we were able to translate this into facts. So we isolate macrophages from the mammary gland, and then the macrophages split really nicely into these CD11B positive and CD11C positive populations. And so we thought that these could be these ductal macrophages, or DMs. And matching the imaging, they're MHC2 high and Lysix-C low. And so we labeled these ones the stromal macrophages, or SMs, and these ones the ductal macrophages. But to confirm that these were the duct-associated macrophages, I did facts of whole glands. This is the frequency of the duct macrophages, and also of fat pads which had cleared epithelium, so they have no ducts left, and there are no ductal macrophages. And when we do an enrichment for ducts, we get an enrichment for these macrophages. So we know now that these macrophages, the CD11C positive, CD11B low, are very specific to the ducts, and they're not present in the stroma. We also found that they are really high for CX3CR1, whereas the stromal macrophages are lower for CX3CR1. And this is just a marker of some macrophage populations, but the reporter mice help us to get a really clear picture of where they are and what they look like. This is looking at the outer layer of the duct with the myoepithelial cells labeled with keratin 5, and this obscures the ductal macrophage network. Whereas looking at the inside of the duct, this network is revealed with the CX3CR1, and then removing the outer layer of the epithelium completely, you can see all of these cells. So they're really, really frequent across the whole gland. So we look at the nipple area, the mid, distal, and in lactation, and they have a really similar distribution across all of these areas. But we really wanted to know what the dynamics of this interaction were. How do the ductal macrophages move, and how do they interact with the epithelium? And to do this, I developed an intravital imaging technique using a skin flap. So we expose the mammary gland, and we can image for up to 12 hours. We have access to an entire mammary gland, which is really important because most of it is embedded in fat tissue, which is really hard to image through. And so having access to the entire gland helps to isolate ducts which don't have much fat on top, and to get really nice resolution. And so this is a movie, intravital movie of the duct, which expresses GFP in purple. And then we have an MHC2 antibody fluorescently conjugated, and we digitally isolate the ductal macrophage signal on the duct using surface rendering and masking. And this showed that the DMs don't migrate around the duct, but they have dendrites that they move back and forth to survey the duct. And I quantified this by tracking the cell body of the ductal macrophages and their dendrites, and also the stromal immune cells, showing that they don't migrate but move their dendrites back and forth. And then looking at how they cover the duct, at any one time they have about 30% coverage of the duct, but through dendrite movement, we can watch the cumulative signal, how it covers the duct, and this coverage increases really rapidly in just a few hours. So it looks like these ductal macrophages can contact almost all epithelial cells in the whole mammary gland within every few hours. So are these cells long-lived resident macrophages? And to answer this question, we use bone marrow chimeras. And this is where we irradiate a mouse to deplete its bone marrow and then inject GFP-positive bone marrow. And we shield the mammary glands to prevent them from being damaged. And then we can look at contribution of the GFP-positive bone marrow cells to different populations. So monocytes are really rapidly replenished, and so we normalize these to 100% replacement. And neutrophils as well are rapidly replenished, so they match the monocytes. Whereas the macrophages in the mammary gland are slowly replaced, so these are long-lived. And so they have a lower rate of replacement by GFP-positive bone marrow. and they have different levels, different rates of replacement. But the ductal macrophages have a half-life of about 5 months, so they are long-lived resident macrophages. But are they initially derived from embryonic development, or do they come later during adult development? And to do this, we use a tool called the MS4A3 lineage tracing. And this was published last year. And it is a really great tool because lineage tracing in macrophages used to be quite difficult. But this mouse, using MS4A3 TD tomato lineage tracing, labels all adult bone marrow-derived macrophages because it's active in the progenitor of the adult macrophages, but not the embryonic macrophages. So what you see here is that at birth, there are no macrophages labeled because they're all embryonic. But then during growth, populations such as microglia, which are embryonic for life, are not contributed to at all by adult bone marrow macrophages. Whereas other populations, such as the colon and the small intestine, are quickly replaced by adult macrophages as the mice grow. And so when we looked at the frequency of the macrophages in early mammary gland development, this is during puberty here, from 4 to 6 weeks, you get a really rapid increase in the ductal macrophages, whereas the other populations are more steady in this time. And then looking at these mice which track the adult macrophages before puberty, only a few of them are adult derived, so most of them are embryonic. But then during this ductal growth, an increase in numbers in DMs, they're coming in from the bone marrow. And then this stabilizes more as they grow older. So initially, ductal macrophages are embryonic, but as the ducts grow and branch during puberty, they come in from the bone marrow. And then to get more of an idea of what these cells might be doing, we did RNA sequencing, and this showed, using this MDS plot here, that the ductal macrophages are really different from the other myeloid cells in the mammary gland. And they have a signature of phagocytic enzymes. And also when we compare the ductal macrophages to the other macrophages, we brought out this notch signaling signature, which is interesting because it has been implicated in mammary gland macrophages before, and also stem cell regulation. And to test this, we had a notch reporter mouse. So this is a notch target gene with the GFP downstream, and it's only active during active notch signaling. And this was specifically on in the ductal macrophages, but not the other immune cells. And so to look at whether notch signaling was important, we looked at CD11C Cree RBPJ flox mice, and this knocks out RBPJ, which is an effector transcription factor of notch signaling, specifically in ductal macrophages. And when we did this, it didn't actually decrease their number significantly, but rather than being CD11B low, they remain CD11B high. So we think that notch might be important for the maturation of ductal macrophages. And it doesn't really affect their localization. They're still within the epithelium. To see what they might be doing during remodeling of the mammary gland, I did FACS across all of these time points through pregnancy, lactation, and involution, and found that this really striking increase in ductal macrophages into involution, where they really dominate the immune landscape. This increase is driven in part by proliferation of the macrophages within the epithelium, not new ones coming in. And so to ask why there might be so many of them here, I did imaging to see where they were, and they're on the outside of the alveoli in lactation, and these are the milk-producing cells here. But at 4 days involution, as the milk-producing cells are dying and the alveoli collapse, The ductal macrophages infiltrate into the lumen and they surround the dying cells. So it looks like they might be phagocytosing these and clearing them away. To get a clearer picture of this, I looked a bit earlier before the alveoli had collapsed. And here the milk-producing cells express the GFP in purple. And looking closer, you can see that these milk-producing cells, which have a slightly dimmer GFP because they're dying, are being engulfed by the ductal macrophages with the MHC2 signal here. And really interestingly, the milk-producing cells are binucleated, so they have these two really distinct nuclei per cell. And within the ductal macrophages, you can see these two nuclei and see that these milk-producing cells are being phagocytosed. But is this important? Are the ductal macrophages necessary for this remodeling? And to answer this, we depleted them using the diphtheria toxin model. So if you express the diphtheria toxin receptor in specific cells, you can administer diphtheria toxin, and this will bind to this receptor and kill just those cells. And so we treated CD11C DTR mice with diphtheria toxin upon weaning, and then collected them during involution. And in the control mice, we have this infiltration of the DMs and phagocytosis of the dying cells. Whereas when we deplete, we have no DMs and we have no phagocytosis of the dying cells. During involution, the lumen collapse and the alveoli collapse to allow remodeling to the normal ducts. And this occurs normally in the controls. But when we deplete the DMs, we get a failure of the lumen to collapse. And so this remodeling is really badly affected. And this is really important for later pregnancies and has implications for cancer as well. So through this discovery, we've really nailed down a population that is important for this remodeling during involution. But there are a lot of other areas that macrophages are important for in mammary gland development that still need further investigation as to which populations are important and what mechanisms. Now, switching tact a bit into breast disease in breast cancer, breast macrophages or tumor-associated macrophages are different to the normal macrophages. So in the normal breast, we have the CD11B positive cells. And as tumors progress, they get more and more of these CD11B low TAMs. So this study separated them into mammary tissue macrophages and tumor-associated macrophages, which has this really interesting phenotype. And so this really piqued our interest because the breast TAMs are pro-tumorigenic, they're notch dependent, and they can proliferate. And so all of these features seem really similar to the ductal macrophages. So we took our RNA-seq and then compared it to RNA-seq of the tumor macrophages, and the genes that were up in the tumor-associated macrophages and the ductal macrophages correlate really strongly, whereas the genes up in the stromal macrophages correlate with the mammary tissue macrophages. And then when we look by flow cytometry, we can see the ductal macrophages here, the CD11C positive, Lysic C low. During tumorigenesis, as the cancer grows, we get enrichment for these cells. And furthermore, they mirror the morphology and positioning of the ductal macrophages in the normal duct within the cancer. So to summarize, the mammary gland has a population of unique resident macrophages that monitor the epithelium. They're bone marrow derived during puberty, but before that, they are embryonic derived. They promote epithelial remodeling by clearing away the dying milk-producing cells during involution. And they're really similar to breast tumor macrophages. And we hope that this discovery will help us to understand better tumor macrophages and help us to modulate those to improve cancer treatment. And so these ductal macrophages, they have a unique markers compared to the stromal macrophages, and they have this intraepithelial localization. During pregnancy, they proliferate to increase in number so that they cover the whole gland and all of the alveoli in lactation. And then in involution, they infiltrate the lumen and clear away these dying milk-producing cells. And then in tumorigenesis, in breast cancer, they maintain this positioning throughout the cancer. So I'd like to thank now, especially my supervisors, Jane Visvader and Rios and Geoff Lindeman, everyone else who was involved in this paper in the lab, Bhupinder, Francois, Gordon Smythe and Luke Gandolfo did the bioinformatic analysis, Felicity Jackling, and everyone else in the lab and the division. I'd like to thank the imaging facility, animal and facts facilities at the Walter and Eliza Hall Institute, at the Taoudi Institute, especially Scott Mueller, who helped with the Intravital imaging and with the project, collaborators at A-STAR in Singapore for the MS4A3 experiments, tissue donors, and funding. And here's Jane and Anne with their 3D glasses, because I do want to quickly show you some cool things you can do with Imaris with the 3D visualization that not a lot of people have seen. And I'd be happy to chat about this with people, and you can check out some of these 3D visualizations here to look at with the Cyan Red 3D glasses. And that's it. Thank you very much for watching, and hope to connect with you in the real world.